Hello everyone. So today we are diving into an intriguing experiment by exploring how temperature affects the cell membrane of the purple beets, also called beta vulgaris, by observing the change in the color of the pigment called beta-lane. So to find out whether a temperature can actually affect the membrane of the cell or not, let's first understand what a cell membrane is. So cell membranes are like the gatekeepers of the cell that controls what goes in and out of the cell. They are made up of different stuff like lipids, cholesterols, and carbohydrates. One crucial part is the phospholipid, which makes a kind of barrier between the cell and its surrounding. This barrier is semi-permeable. That means some stuff can pass through it while other cannot. So this is impacted by a lot of factors such as temperature, medium osmolarity, and the type of solute passing the membrane. So as I said, that temperature affects the stability and permeability of the membrane. So temperature plays a big role how well the barrier, like the semi-permeable barrier, works. So if the temperature changes, it can mess up with the cell membrane's job. For example, when it gets hot, the membrane becomes more fluid, and this allows things to pass through it more easily. But when it's cold, the membrane becomes stiff and it becomes harder for things to get through it. So think of it like this. At higher temperature, the membrane becomes more fluid, while at lower temperature, it becomes more rigid. So how we are going to test this? Well, first, we'll need some purple beets. We'll cut them into equal pieces and set up water baths at different temperatures, like 35 degrees Celsius, 50 degrees Celsius, 65 degree, as well as ice baths. And we'll also take one at the room temperature. Then we'll incubate our beet samples at different temperatures for 30 minutes. Once our beet has been incubated, it's time to measure the absorbance of the beet pigment using spectrophotometer at a wavelength of 510 nanometer. This will give us an idea of how much pigment has leaked out of the cell, indicating membrane permeability. We'll record these observation values and calculate the average for each temperature. And at the end, to ensure the reliability of our result, we'll repeat the experiment one more time. This way we can confirm that our findings are consistent and accurate. So, what we can conclude from this experiment is that temperature plays a significant role in the stability and permeability of the cell membranes. By investigating its effect on the pigment leakage of purple beets, we can gain valuable insight into how cell responds to changes in their environment.
So in this slide, I would like to explain about the result and interpretation of our experiment. So the main objective was to investigate that how the temperature change can affect the stability and permeability of the cell membrane of Vita vulgaris, that is beetroot. So the key findings of our experiment showed us that at low temperature membrane fluidity becomes more rigid, which ultimately causes the beetroot to become less permeable. So on the other hand, if we talk about at higher temperatures, at higher temperature, the membrane becomes more unstable, which means that pigment leakage is more, which makes the membrane more fluid and permeable. So the next slide, we will describe the further part. So we can clearly see from the graphs that at higher temperature, we performed our experiment twice, week 1 and week 2, and both the weeks the highest absorbance value was at 65 degrees Celsius. So if the absorbance value is more, it means the pigment leakage is also more. So we can clearly find out from this that if the pigment leakage is more, that means the membrane has become more destabilized and it has become more fluid. So all these figures demonstrates the change in the integrity and stability of the cell membrane. So we can clearly compare our result with all the temperatures, that is ice bath room temperature 35 degree and 50 degrees, that 65 degrees Celsius the beetroot had more pigment leakage in comparison to all these temperatures. So we can also see if here that the color of the third tube is more vibrant and dark, which clearly tells that the pigment leakage was more at 65 degrees Celsius. We can look from the color of the pigment with the varying temperatures. So to conclude that I would say that at lower temperature preserves the membrane integrity more fully than the higher temperature, which is directly linked to the pigment leakage membrane destabilization. So this is all about our pigment leakage. So stability and selectivity of membrane is also enhanced by the various molecules which are present inside the membrane, that are proteins, lipids, and carbohydrates. Integrity and stability of the membrane was highly influenced at 65 degrees Celsius, and the lowest was at ice bath. So these are our results. So these were the references we took for our experiment. Thank you.